So welcome everyone to Strait Science. Strait Science is an evening seminar series put on by University of Alaska Fairbanks, UAF, Northwest Campus, right here in Nome, Alaska, and UAF Alaska Sea Grant. So UAF Sea Grant and UAF Northwest Campus, we are public servants of the Bering Strait region. We serve all peoples of the Bering Strait region, which is the homeland and waters of the Inupiaq, Yupik, and St. Lawrence Island Yupik peoples. So tonight we've got Jiaxu Zhang from the University of Washington and the NOAA Pacific Marine Environmental Lab. She's going to start us off, and Mike Steele, who is with the University of Washington Applied Physics Lab. What are they going to talk about? They're going to talk about harmful algal bloom research, which is a topic that is of big interest for us here in the Bering Strait region, since during 2022, we had the nation's largest, most persistent, and most toxic bloom of an algae called Alexandrium that produces a poison called saxitoxin that can give warm-blooded animals, seabirds, human beings, and other creatures problems, sickness called paralytic shellfish poisoning. And since we won the prize, and we haven't had to deal with this issue before, it's really wonderful that we have so much research, both at the local level and also with people coming from afar to tackle this. This has not gone unnoticed what happened here in 2022. And as we all know, it takes research a little bit of time to get going, but they're here now. And so both Mike Steele, who is currently in Seattle, so he's on a different time zone, and Jiaxu from Seattle, but she's here with us tonight, and her pilots are here tonight as well. And she will tell us what she's doing with aircraft and how this may help us understand and maybe detect harmful algal blooms. So we're so glad you're with us. Take it away.
Thank you, Gay, for your introduction. So before I get started, I want to turn my camera to my crew member who is sitting here with me in the room. So
you guys all wave there.
Yeah, so we have Lieutenant Mason Carroll, Lieutenant Chelsea Parrish, and our mechanics Ron Polly. Yep. So if you have any aircraft related questions, you can bring them up later after the talk. So for this talk, I want to start with a very quick background introduction of the phytoplankton and the harmful algae blooms. So the phytoplankton is the small tiny plants living at the ocean surface, and they produce the food, form the base of marine food webs. So the smaller sized phytoplankton, usually they tend to be floating at the ocean surface, and they will be eaten by the zooplankton, and then to the small fish, and then to the larger sized fish. While on the other hand, the fairly large sized phytoplankton, usually they can form a chain, and then it's too heavy for them to stay on the surface, and then they will sink into the ocean bottom. And that will provide some direct food to the benthic biomass, benthic species. So here on the map, I'm not sure if you can see my mouse. That's perfect. So on the right is a map of our region, and then with all the colored dots showing our coastal community community. So here, actually, for example, this region to the south of the Daimi Islands, and then here to the west of the Point Hope, there are a few regions that the large-sized phytoplankton will sink to the bottom and start to feed the benthos. So, however, some of the species, they can form harmful algae blooms. And the issue is that here in our region, it used to be so cold, so this HABs is not a big issue until recently. It's getting warmer and warmer, So we see a increased trend in this region, in this Pacific Arctic region of the HAB events. A profound example is the event in August 2022, which is one of the most vast and toxic Alexandrium blooms ever documented globally. So the figure on the right, lower right, shows the Alexandrium catenella cell density as measured by one of the research vessels, Norseman 2, by their NOAA instrument. So the color shows here, we see like the red color is actually over 60,000 cells per liter, which is 100 times higher than the level needed to trigger public health warnings. So with this information, the question we raise is that we were lucky to have Norseman 2 at that time so that they can take NOAA measurements. What if there is no ship around at the time of the HAB events? Is there some other way that we can see some early signs for this HAB to show up? So the answer is yes. The answer is the ocean color remote sensing. So the way we do it is to use the satellite images, the data that collected by the ocean color sensors that is mounted on the satellite. So the idea behind that is because the phytoplankton, they absorb blue and white light and reflect more green lights. So that's why we see all the plants have the green leaves. So the sensor just detect the amount of the sunlight reflected from the ocean surface and calculate the ratio between the blue and the green light, and then detect like how much has been absorbed, down there by the phytoplankton. So that's the idea behind it. There is some other information we can track from this remote sensing images. For example, what's the size distribution of this phytoplankton, whether it's small versus large, or even sometimes there is very specific algorithms that can be developed to detect harmful species. So our group is one of the few groups that work on harmful species algorithm in this region. So the figure on the upper right is an enhanced RGB figure, which has been captured. There happened to be a very beautiful day in August in 2022, and we get this image in the Bering Strait, and we saw this brown color, a dragon shape pattern over there. And based on our algorithm, which is on the lower right, we see it is a Alexandrium blue. So that this have been verified by the Norseman 2 NOAA detection. So, well, this is wonderful. This is fabulous. But you may think, is this enough? If this is good, why do we need other thing? We can rely on satellite all the time and go from there. But the answer is actually satellite have other limitations. For example, most of them are very low in resolution. 1 pixel is about like 300 meter to 1 kilometer. Many of them, they make less frequent revisit. So they come, basically you don't have a daily coverage. Some of them come like every month with the really high resolution ones. Another issue here is the persistent cloud cover during the summertime, especially towards the late of the summer, like August, September. That's actually the peak season for the harmful algae bloom events to occur in this region. And another very big issue is the limited spectral bands. So I'll get into this point later, but the idea is basically we have very big uncertainty of all the algorithm which exist at this moment. So to better explain what it means, I want to introduce a new satellite, which is called PACE, P-A-C-E, stands for Phytoplankton, Aerosol, Cloud, Ocean Ecosystem. So this mission has been launched early this year in February, and the figure on the right shows what it looks like in the space. So the major difference, as I mentioned previously, is the multispectral versus hyperspectral. So the figure on the lower left shows that because we see the spectral from the blue color purple color, all the way to the red, infrared. So the rainbow color actually, all the previous satellites, they only take one cut at some of the bands. And then, for example, here, it's just like a example. So you see there are only 6 points basically on the band. So you only see like, bar plot. It is very limited information. However, now we have a much powerful satellite which takes like 300 images along on this spectral, and on the right you see a continuous spectral curve that gives us a much more information that can significantly increase the differentiation among the phytoplankton groups. So the figure on the right actually shows jars with the phytoplankton cultures in it. And you can see that actually different species have very different colors, light green, blue green, light green, dark green, brown, some are even pinkish. So actually all this information can reflect on the spectral plot like very differently. So you can use this enhanced information to do that. So now you say, okay, that's fabulous. We have pace in the sky. Are we all done? Are we all set? Unfortunately, it's not, because we still have the cover issue. We still have the resolution issue. We have other issues. So the idea that we want to do is, let's take this idea, the hyperspectral idea, but put it in an aircraft so that we can fly much lower. We can get high resolution data, and we can fly below some cloud covers so that we can get data when the satellite cannot. So this is where we get in. So who we are, our program is called Arctic Air. Air stands for Airborne Investigations and Research. So we are a research program at NOAA's Pacific Marine Environmental Lab, PMEL, in Seattle. And our mission is to use aircraft and UAVs to study physical, chemical, and biological processes in the Pacific Arctic, especially at times and scales otherwise unavailable to researchers. So we collaborate among NOAA, among the line offices, and we work with other government agencies, and we also have a lot of collaborations with the academia. So the figure in the middle is the Twin Otter aircraft. So actually, which is in the airport. We just jumped out of the aircraft an hour ago and came here to give the presentation. On the right is a free fly Alta X, which is a drone model that we're going to use. I'll talk about that later. So the way we do it is to use a resonant PICA-L hyperspectral imaging system. We have a photo of that PICA-L on the right. So what we do is that we mount it at the belly of the aircraft. When we fly over, we're collecting the so-called data cube along the way, and then we get the hyperspectral data cube over the swaths that we fly over. And after that, we do a whole bunch of atmospheric correction, geo-rectification, radiance to reflectance conversion, apply a whole bunch of algorithm to smooth the data, to correct the sun glint effect, and then finally get the curve that we use to interpret it, to apply all the algorithm. So we started this practice in 2023 in June. So we got the camera, we install it on the aircraft. We take some training, and in June that's the first time we fly with it. So on the right you can see the flight path is like a busy bee going everywhere. Actually, we were testing like different altitude, different directions, different positions, try to get some sample data to play with. And at that time, we also deployed 6 micro swift TS wave buoys which also measure temperature salinity along the DBO3 line, which is right to the west of the Point Hope, somewhere here. Sorry, my mouse moves very slowly on the screen. And then, so Mike will talk about that later. So later in September, we come back again. We start to try to coordinate with a research vessel, Sikuliak, and also we are trying to do some overflights with some mooring system. So on the lower right is a mooring system over the Kosovo area. So we did some like experiments, basically, try to collect some data. So that was in 2023. And in all this sample data, there was one day which was really clear. We got a very nice image at along DBO3. So on the left, we see a very clear bloom happening on here. Because it is so high resolution, we even see a very thin line of foam, which often occur on the edge of a bloom. So, and then we apply.
Can I ask a question?
Oh, yeah.
Can you define what a bloom is?
So from this, yes, good question. So we did some calculation, and based on what we're not sure at that time, we think it's a diatom bloom, which is a healthy bloom, but we are not sure. Luckily, 3 days later, Sikuliak went by, and their IFCB detected, confirmed that it is a diatom bloom.
But a bloom is a uncontrollable growth of algae, just because people in the audience may not know that, be familiar with the term bloom.
Mm-hmm. Yeah, so I assume that the definition of the bloom mostly based on the concentration of the cell. So when the cell exceeds certain number, we consider it is a bloom. So it could be a healthy bloom, which is a happy thing, but it could also be a harmful bloom, which produce a lot of toxins that can further pass to the the shellfish, the fish, and to the ecosystem. So
and
that's
it depends on the type of algae that's growing so fast it creates a bloom. That's
exactly
what I think you're referring to. Good algae
versus
bloom,
the
which we need, right? To breathe oxygen, we need these algae and to feed everything. But
Thank
the
you.
harmful species are the tough ones we're experiencing in 2022 and 2023.
Yes.
New to us. Thank you. In that quantity.
Mm-hmm.
Thank you.
Thank you. Thank you for jumping in and explaining that. Exactly. So yeah, we did a bunch of analysis. You know, we think this it's definitely it's a proof that this idea actually works. So it's really like a proof of our assumption. So so we continued this research. So in a few months ago, we started this year's survey. So in June, the weather was really good. So we were able to collect really nice data. So on the right, I'm showing all the curves. So the red curves actually are all the data that we collected on very good weather conditions. So this data, we're still working on that. And meanwhile, we deployed 6 buoys here. So Mike will talk about these buoys later. And while we were working on this, we find that on that day, actually, we overlaid this flat path on a satellite image. And actually, this brownish color is a algae bloom that was occurring exactly at the same time when we flew over. And no alarm. This is a healthy one. It should be a diatom bloom, which is very typical at this time of the year. So yeah, so we're still working on that. Hopefully, we can find. There's also sea ice to the north of it. So we're trying to combine the buoy data, the images, the flight data, try to understand some sea ice, ocean, phytoplankton interaction during that time of the year. So we're working on that. So currently we're here to do a survey flight in September. So so for this survey, we actually started from Anchorage, which is usually where the the aircraft will be configured for the summer field season. And then we flew all the way to the north to Prudhoe Bay and deploy a whole bunch of buoys to the north of Dead Horse and also the Barrow area, the point of Barrow. And then we come down to Nome and started our hyperspectral data collection. So here, so this is what we typically do. So the day before, I just check some available, because it's so cloudy. So actually, it's very hard to get a good image. But I check a few days ago, for example, to see whether there is a bloom or not, and then check the weather forecast, check some, and discuss the flight plan with our pilots. So actually, they can help address some, like how to make that happen if the audience are interested in learning that. And then we fly over, we go there, try to find some good light condition for the camera to work, and then just to collect data. Ideally, we want to fly north and south so that we can avoid the sun glint. If you're facing the sun or go to the other way, you don't have the glint effect at the surface. But if you like from east to west, very likely you will have a very glint at the image, which is like a big bright, like a area on the image, which is something we don't really like. So and then on the map, you notice there are some yellow small dots. Actually, they are the currently there is a ship. It's called Northwest Explorer. It is doing it's working for NOAA, doing a surface trial survey in the northern Bering Sea. So their major target is to survey all the fish, like the different species, the counts, the size, weight, and also they collect the water samples, which actually the chief scientist is holding. In the little jar, there are our beloved phytoplankton, and all these jars will be sent to the Algal lab and then go through the microscopy and other like IFCB to do the speciation and count for each of them. So the reason why we overfly them is that we need some ground truthing so that can verify what we find up in the sky. Remote sensing can always be wrong, but if we have some samples at the ocean surface that can help verify what we see, that will greatly improve our algorithm in the future. Besides on that, we also deploy buoys from the twin otter. So on the figure to the left, you see there is a little tip at the belly. That is the tube that we deploy the buoys. And this time we deployed a few so-called AXBT that just deploy at one location, but they sink all the way down to about 1,000-meter depths. And then they measure the ocean temperature. And we also deploy the MicroSwift TS buoys, which is to the right. And I'm going to turn to Mike and ask him to talk about all these exciting buoys. Do I stop presenting?
You can just, if he comes on, he's got a video and unmute himself. And then you can just turn your video off until he's...
Do I have to stop sharing?
No,
I guess so.
I'm not sure how you guys have that working.
I've got my
I'm going to
audio.
stop sharing.
Okay.
It looks
You can
like
go. You can go visual.
it says unable to start video.
This I do not know. I did not know you were going to show a video. So
No,
there
no.
may
I
be
mean,
some...
just my. You've it says host disabled participant screen sharing, so you got to let me share.
Oh, thank you. Yeah, hang on. I didn't think of you as a... I didn't know you were going to be part of the thing. So hang on. We'll get that rectified. Thank you.
No worries.
Okay. You should be a co-host now.
Yes.
So now you should share your screen. Thank you.
Yeah. Okay. Can you see my screen?
We can.
Yep.
Okay, good. All right, I'll go into presenter mode here. Here we go. Okay, so thank you. Thank you for the invitation. I'm sorry I can't be there, but I'm there in spirit. And I was there in June. And I'm going to talk about these buoys that we're deploying. So deploying by aircraft, ocean.
Mike, can you hold on for a sec? Actually, your screen is not on the screen in the room.
It is for everyone, but you need to go now back to the Zoom meeting because you're in this.
Oh, so can I just
Yeah,
pull
just
that?
minimize it for a second, and you can, or you can come over here and watch.
Yeah, actually, I want everyone else to see that.
No, everyone else is seeing it.
Yeah, but not in this room.
Yes, correct. So
we can see
Yeah,
the
let me-
screen.
yes, there's some people. Thank you, Cheryl. There's some people in here that want to see. And I forgot because I've got that computer. Can you minimize? Thank you, Mike, for your patience. Hang on.
No worries. Let me know when you're ready.
Yeah, just hold on for a sec. Let me exit this so that I can drag that Zoom.
Your screen.
Yeah,
There we
to
go.
here. It's not super big.
Thank you, everybody online. We're just getting it so people in the room can see what you're seeing too, because we're switching things. And we got it. All right. Thank you, Mike.
Okay, ready?
We are ready.
Okay, here we go. Okay, so all right, I'm talking about buoys, air deployable buoys for Arctic research. I'm Mike Steele. I'm the guy who's looking at ocean temperature and salinity, and my collaborator is Jim Thompson, who is an ocean surface wave guy. We both work at the Applied Physics Lab at the University of Washington in Seattle. So a little background. Everybody knows sea ice is melting in the Arctic. I was in Boise, Idaho, just a few weeks ago. And so, it's like a little more than a third of an Idaho per year of ice loss over the whole Arctic. But the blue line shows that over the whole Arctic, there was like 7.5 million square kilometers of ice in September, the end of summer, in the old days. And now it's like 4.5. So, a lot less. There's been these spectacular dips in the time series, and the first big dip was 2007 when we lost a bunch of ice. And so a bunch of people wrote papers about that. I wrote a paper about how when the ice goes away, guess what? The sun can warm up the water. It's, not brain surgery, but it's new for the Arctic, or relatively new. And so I was pretty much the first guy into that field. It's like ice is going away, the ocean's warming up. And so that's been a big part of my career in recent years. Now, this figure was made using satellite data. There's satellites that basically have infrared cameras that are measuring, taking pictures in the infrared of the temperature of the Earth's surface, and those are great. But Jiaxu was also telling you about how satellites are great, but not so great. The resolution isn't awesome, and maybe you don't trust them. You want to actually make a measurement in the water. In this case, just a thermometer in the water to make sure the satellite's getting the right number. And then maybe you want to take a measurement below the surface as well. So motivated by that, I started making buoys. And so my older project, which is continuing to this day, I fly with a Coast Guard based out of Kodiak in a project called Seasonal Ice Zone Reconnaissance Surveys. And we fly clear across Alaska to the Beaufort Sea and drop big buoys out of this big plane. And the buoys are great. They got lots of sensors. They have a long life of several years, and they cost a lot of money. So that's sort of frustrating for me. And Jiaxu's program, Arctic Air, is a wonderful thing. I love it because I get to fly in a much more fun airplane, a Twin Otter, that's smaller, more nimble, and my buoys are smaller and cheaper. So it's a great opportunity. So I want to tell you about this buoy called a microswift buoy, but I have to start with a not micro, but just the swift buoy that Jim Thompson developed. It stands for a surface wave instrument float with tracking. So it's basically a big expensive wave buoy with some other stuff on it. So Jim, he's got a big buoy. I've got a big buoy to measure temperature and salinity. And we were both frustrated at the same time. Hello? Yeah, okay. We were both frustrated at the same time with our big expensive buoys, and we both were thinking about making smaller ones. So we have collaborated on this buoy called a micro swift buoy. So it's much smaller than any of our big buoys. It's got two antennae in it, Iridium for satellite communication and GPS for position. It's got nice, friendly alkaline batteries, no hazmat there. And it's measuring surface waves, SST, that's sea surface temperature, and SSS, that's sea surface salinity, how salty the ocean is at the surface. At this point, we've got a 2-month lifetime. And for air deployment, they come packaged in a cardboard tube with a parachute in the top. And that red thing on the bottom is a magnet that helps me turn the buoy on, and then I bring it home. So this is what the inside of the Twin Otter looks like. There's Jiaxu and me near the front. And there's this was from June of this year. That's Phil, one of the buoy engineers, in the back. And then I know the pilots are there. I know it's not called a buoy room, but I'm going to call it a buoy room for my purposes in the back. And so in the back, there's a little room there where we can get the buoys ready to be deployed. And there's a tube, a hole in the bottom of the plane where we can push the buoys out when we get to the position we want to be at. So I'm going to show you a movie, hopefully this works, of a deployment of one of these buoys out the tube. So there's a GoPro camera looking back underneath the plane. This was in June this year, west of Nome. There was some ice, especially in the western part, like towards Russia. And let's go. And you'll see the buoy come out the tube and the blue parachute get deployed. So come on. Here we go. And there it is. And there it goes. Okay. So I have a website. Anyone can go there. It's called the Uptempo website. That was the original name of my program. Maybe I'll change it in the future. I called it Upper Temperature of the Polar Oceans, Uptempo. Temperature measurements are cheap. Thermometers are cheap. Salinity, measuring how salty the ocean is, more expensive. So I started with temperature, but I'm getting into salinity as well. So there's the website address. And here is the kind of picture you can get from the website. So here it was on June 2nd of this year. We deployed a cluster of 6 buoys west of Nome. So the colors show you the sea surface temperature. Red is warm, blue is cold. This is from satellite. And then the white and grayscale is the amount of ice, of sea ice floating on the ocean. And I know it's all clustered up there on the top. It's a big picture because we've got buoys all over the Arctic Ocean, as you can see. And then 2 months later, at the end of July, we had 4 buoys left. You can see they got distributed out a little bit. 2 of the buoys died like within a few days or a week. I think they got squished by ice. 2 made it north through Bering Strait. 3 sampled sort of western Norton Sound. 1 made a nice circuit of Norton Sound. I'll show you in a minute. And when you go to my website, you can click, and you can click through week by week over the last like 10 years of Arctic temperatures and ice. So it's a fun tool to use. Here's the buoy that went through, made a circuit of Norton Sound. So you see the start on the left, and then it kind of headed east into the sound, and then ended where that red flag is there. Here's the time series of sea surface temperature and sea surface salinity from that buoy. So from early June to the end of July, and these are temperatures that are in Celsius that are hotter than I'm used to seeing, and because I generally work in the sort of the high Arctic, like north of Bering Strait, and quite fresh values as well. I feel like I'm describing a cinnamon roll here, hot and fresh, but and what's interesting is kind of in the middle of the time series, there's this really interesting excursion where the temperatures got very warm, the salinity got very low, very fresh. And that's a signature of the ACC, the Alaska Coastal Current, that rides northward along the shelf break. And you can even see the buoy was deflected a little bit as it transited that. I'd love to do more work based out of Nome and really sample this very, very interesting environment, sort of the open ocean to the west, Norton Sound to the east, and the this strong current in the middle. So in summary, we've deployed buoys 4 times now, in June and September last year, and June and September this year, based out of a number of places, including Nome in June. These colors in this case are showing sea surface temperature. So we've got some areas that are, you know, dark blue, that's nice and cold, and then other areas that are quite warm, and some nice temperature fronts there as well. So in summary, for the future, what do we want to do? Well, we definitely have to integrate more with the kind of ocean color observations that Jiaxu was talking about, you know, warm waters enhancing these blooms. We've got to better integrate with these ocean color observations. I have a postdoc who we've written a paper. We have digitized the sea ice observations from the Anchorage National Weather Service, the sea ice desk there. And it's a really wonderful data set, and our digitized version of it will be available very soon for public use. And you know, as a matter of course, I always do a comparison with the satellite temperature and salinity observations. I've got an intern, undergraduate intern from the Naval Academy in Annapolis working on that with me right now. Now, we're also interested in putting more sensors on these little micro buoys. And even maybe a string of sensors for subsurface observations. You might call that mission creep, you know, like, you know, just load this little thing up with lots of sensors. And maybe that's kind of counterproductive to our idea of, you know, small and cheap. So this is a balancing act we'll have to work with. I have some funding now, and we're working with some clever materials science engineers to make our buoy hulls out of algae. You know, not toxic algae, but good algae. So, you know, bio hulls. We're trying to make our buoys more eco-friendly. And then we have a proposal into the the North Pacific Research Board, NPRB, working with Jiaxu to hopefully do more work in the Northern Bering Sea. And with that, I am done. Thank you very much.
Hello.
Thank
Hi.
you, Mike.
I have a question.
Yeah, question?
Go ahead. Go ahead, Edwin. Go ahead.
You're muted.
Oh, okay, there it is. Yeah, I wanted to ask about the buoy. The current was taken from the ACC, I think that you were talking about, and
Yes.
so it went eastwards into the Norton Sound and up towards Nome.
It did. The current I'm talking about runs from the south to the north along the shelf break, that arrow that I've got there in the figure.
Okay, so are you gonna be posting your site or email like you said, so that we could look through the?
Well, I'm happy for yeah, I mean I'm definitely happy to give my slides to Gay or whoever, and you're welcome to look through them, but let's go back again. Let's
So
go
Mike,
back here.
Mike?
That's my website. Yeah.
Yeah, this is Gay. If you could just open your website and then put the link in the chat, everyone that's online can just click on it.
Oh,
As
yeah, I can
opposed
totally do that.
writing down here.
Oh yeah, I'd be happy to do that. And here's the
There you
slide.
go.
But
Yep.
yeah, I'll be happy to put it in the chat.
And then if you could go back to that slide that Edwin was talking about where you have because that's of interest, the start and end. So was this a time, because you were flying out and dropping your buoy out the plane west of Nome? So that is where it started. Was there a lot of north wind?
Yes.
Is that
You remember
so?
all the buoys? So we put it in a line, you know, trending sort of east-west line, and sort of the climatological ocean currents all moved northward. We expected them all to just make a beeline northward into Bering Strait, but the winds were screaming out of the north, and they all went straight. They all tried to die on the north coast of St. Lawrence Island, basically. But luckily they did.
one of the buoys actually was reported, and I think you or Jim Thompson, your other researcher, contacted me, and
Yes.
because you wanted to recover the buoys if possible, and it looked like they had landed at Savunga. This trail you show on the green is just one of the buoys, but
That's
you
right.
had several buoys. Yeah.
That's right.
Yeah. So on that one, there was a lot of interest in trying to help you get your buoy back.
Yes.
And one buoy was actually photographed up at just south of near Tin City on a beach. And is there, if people see these buoys, this was an issue that came up, and I think Jim Thompson was very generous. Is there, if people still see your buoys, I don't know if you're expecting them back, or you know where they are, or you've recovered them all, or they're not to be recovered. I don't know what happens to them. But if people see your buoy, there is information on who to contact.
Yes.
If people, okay. And
Yeah.
then is there any kind of reward? Because that was a question. There was a reward at the time. Is there a reward still if someone were to find one of your buoys?
Yes.
Okay, so that's great. And maybe you can click back to a picture of the buoy.
Yes. I mean, I'd like to work with you and, get a, get that codified in some
There you
way. I don't know, out to the
Yeah.
community. Yeah.
Yeah, and so it's what is it, half a meter? So what's that, like 2 feet?
Couple of
Foot
feet.
and a half.
Yeah.
Okay. And that's what it looks like. And one of these was photographed, and I got a picture of it, and the gentleman wasn't sure if it had any kind of toxic whatever. And
Nope.
but he did get in touch. It does not have any hazardous materials. And so I don't know, that was between him and Jim Thompson, your partner researcher, whether or not he was able to recover that buoy. I got the two of them able to communicate, and I don't know the outcome of that. But I just thought I'd make a note of that because people here find all kinds of things. I also would recommend you might want to put an American flag on your piece of equipment as
Yeah.
well.
Yeah.
It is in this region, especially nowadays, that is something you really want to make sure we understand. The citizenry here understands who owns this buoy or where it comes from, because we have different things we're supposed to do if we see other things.
Okay. Yeah, great.
So just something to put on, just put a sticker on there or something.
Last
Says
year,
made in
last year, we deployed a buoy pretty close to Utqiagvik, and like within a day or two, it washed up on the beach. Someone found it and shipped it back to Seattle, which was great. And so
Very
yeah, we would love that.
So Mike, I have a few more slides to continue. So if you can stop sharing so that I can
Yes.
start.
Yeah, sorry about that.
Thank you so much. All right, so let me share from my side and hopefully to wrap it up. All right, so let me go back to the slides I started and pick up from there. So I hope the audience already get a general idea like what we're doing, and just bear with me. Let me just.
It's all to try to understand what the satellite is finding. Right?
Yeah, so I hope you still see my screen.
We do. We see you.
Okay,
Perfect.
so currently, so actually, this is our Arctic Air's vision. So we're trying to build a multi-platform observational network. So each platform, it will have its limitation, but it will have its specialty. Currently, we have aircraft in place, we have the paid satellite in place, and we have the surface buoys in place. And in the future, we want to collaborate more with existing research vehicle service. The research vessel, and onboard more instruments that can do more measurements, not only the water sample, but also other hyperspectral measurements near the ocean surface. And we're going to put a UAV based camera, so mount the same camera but under the UAV, and this UAV will go with the ship, and whenever the ship stops, do the station, and then the UAV can fly around and collect data. We also plan to have the autonomous surface vehicles, the ASV, and then we can mount CTDs and IFCB, the imaging flow cytobot that can measure the phytoplankton. And also, we would like to ask the community to use their community vessel-based measurements to help our entire observational network. So here a little bit more about the cruises that we will be working on. So here the star shows the mooring sites in the Northern Bering Sea, and the red stations are the Northern Bering Sea Surface Trial Survey, which we are working right now, but we will going to collaborate in the future. There is also the green dots are the DBO stations that the Arctic cruise, which is usually on Sikiliak. So they will be sampling the DBO1 region, which is to the left, and DBO2 region, which is between St. Lawrence Island and the Northern Bering Sea. And then we will also have the blue dots, which is the spring or fall mooring of the EcoFlowSite program. And we will basically, try to collaborate whoever will be working here to mount this. So one thing that the red dot station will be carrying is as I said before, the FreeFly Alta X. So hopefully we are going to launch it next year on the ship. And then another device we're very excited about is called DALAC. I'm not sure if this is the right pronunciation, but basically it's a hyperspectral device that is mounted to a pole, which will be mounted on the ship and then measure just the, during the transition. So that will be everything autonomous, but that would be very fancy, very nice to have. So actually, I put the wrong year. It should be 2026. So PMEL is purchasing a C-Track. So it's a autonomous surface vehicle, which you see that the yellow boat with the solar panel, so it can move around. So that is all, in our picture. So as I mentioned before, there is a very important component for this observational network is the community engagement. So we do hope that we can rely, whenever we see some early signs, either from the satellite or from the aircraft or from whichever platform, we do need samples to confirm this information. But sometimes, the research vessel is not nearby, the service buoy is not nearby, but we do have fishing boats, local boats for transportation and other things. So we do need your help. It's not that fancy. You don't really need a fancy research vessel to collect these samples. So what you need is just a fishing boat or any small boat. You just go out and pull the ocean water. There's definitely some protocols. I hope Gay and Emma Pate will be able to help, do some very simple training. And then you just, get a jar of the ocean water and send it to Emma and Gay, and then they can ship it to one of our research labs so that we can do analysis. But all the details, we will work on that, but we do hope that the community can help us with that. And meantime, whenever we're here in town, we are doing some community engagement. For example, we provide tours of the research aircraft. We invite some guest observers. Actually, Gay just she flew with us yesterday. Very, very nice flight. And then we talked to Ben from KNOM, and we got interview from the NOAA Nuggets. So all that kind of things. And also we want to establish partnership with local residents. And then we want to co-develop all the research proposals and the research projects. So there are a lot of future opportunities exist. So I just want to get the words out, and whoever want to organize, for example, you have a group of high school students who want to take a tour of that research aircraft, just let me know. So I will be happy to coordinate that. Or the class visits or some other things, I'm all ready and happy to provide that. So that's actually the last slide of my talk, and I'm happy to take any questions if there's any, and also Mike too.
All right. So thank you. And we want to thank Jiaxu and Mike Steele. So while you're thinking of your questions, while you're thinking of your questions, this is a good time to give a little love in the chat box to Mike and Jiaxu, because it's never easy to be a public speaker, and you had a beautiful presentation.
Thank you.
Yeah. So I know, I've got some questions myself. And there was a question in the chat also. Dean Stockwell in Fairbanks was wanting to know, you probably couldn't see this because this came in direct.
No, I cannot see that.
No, that's fine. I've got this. And so he was wanting to know, in your presentation, can you relate some of what you're looking for with your hyperspectral camera? And Dean, let me know if I have this right or wrong, to chlorophyll concentrations.
Yes. So chlorophyll concentration, we can definitely get that information out of the hyperspectral. Basically, all other previous data, all other previous remote sensing products, we can get out of that. And on top of that, we have other information that previous remote sensing cannot get. So yes, chlorophyll concentration, that's basically a ratio between the blue light and the green light. And then with some other parameters, calculations, filters, that sort of thing. And from the hyperspectral that is available, if you go to any of the NASA website or any other EarthView website, you can easily pull that chlorophyll concentration information from all the satellites. Currently, there is VIIRS, there's Sentinel-3 and Sentinel-2, and there's also PACE data that are all available. I hope that answers your question.
Yeah, Dean, I hope I got that right, that question asked correctly.
Yes, you did, but I have another one.
Go ahead.
Many distributions of chlorophyll A in this area, these regions, show chlorophyll maxes at 20 plus 30 meters, and you're looking at surface penetration. What is the depth of penetration that you're looking at?
That's a good question. That's something that I'm not very sure about, because I mostly work on the data collection side, but not too much on the data analysis side. But actually, we're looking at a very shallow depths of the surface. So we can miss some phytoplankton that not live right at the ocean surface, but at a deeper depths. That's something that we can miss for sure.
Yes, most of the chlorophyll A distributions are less than 5 meters. So if you have blooms that are occurring, for example, the ice edge blooms that occur typically occur below that freshwater lens. So you're missing those numbers.
That is correct.
Yeah, you mentioned that this was one of the biggest blooms of Alexandrium recorded, but at the same time, Alexandrium has been recorded throughout the Bering Sea way back to when sampling started. And so I didn't see a lot of deaths of animals occurring, or birds or fish. And so the impact, when people talk about the magnitudes of these things, I start feeling a little bit uneasy, because to me, you have three different cycles of things happening. You have an ice edge bloom where all the ice algae is falling into the water, feeding the benthos. A little bit later, you have an actual phytoplankton bloom that's typical spharedrip type blooms. And then you have these odd other blooms. And sometimes when we're trying to get a distinction between these, everybody's focusing on, oh, this is harmful. This is what we should be studying. But yet, if you don't have that ice algae bloom taking place, you're not feeding the benthos. And so I think you're doing great work. I think it needs to be done. I'm not putting it down, but we have to know the limitations of each step we take. Thank you.
So thanks, Dean. And on that, for the impacts regionally during the 2022, I think the biggest problem was we did not have, as all water rolls north, we really did not have a good understanding of impacts. We did know that at the same time that I think it was August 21st or 22nd, 2022, I did fly from Diomede with the Coast Guard to Kotzebue, and we had, I think it was over 30 dead walrus and 2 dead gray whale, both of which would be what you would not want to see or what you would expect to see dead if they had had harmful algal poisoning. We also had bird die-offs, but that's compounded by the fact that we're have had 8 or 9 years consecutive starvation event because of our transitioning ecosystem. Same with the marine mammals, right? We were in an unusual mortality event up until I think they finally canceled it just recently. Starvation due to our ecosystem transitioning. So impacts, I think you're right. We didn't have a good handle on that, but I do think there is we would have likely missed impacts from that, at least as the water rolled further north, as the coast rolls away, right? The Seward Peninsula and the Chukotka Peninsula are just opening up, and we have a lot of open ocean there. But where we did fly, there was quite a number of dead animals at the time, marine mammals, walrus and gray whale, between Wales and Cape Espenberg on our way to Kotzebue. Unfortunately, we were low on gas, and a piece of the helicopter actually kind of fell off, and so we had to hurry home. So that was frustrating.
Was that the biggest event? Was that the biggest mortality event you ever saw up there?
That we heard of, and it was also our only flight.
No, I mean historically, was that the biggest mortality event up there you saw with dead walruses? And
the other mortality event of that same magnitude was when in 2017, Diomede had also harvested a walrus. So we know what killed it. It was harvested. However, it was not acting in a normal fashion, and the community realized there was something wrong with the animal in its behavior. They called and asked, what should we sample? And we just asked for the normal suite of things. We were sort of thinking, well, we should test it for harmful algal blooms. And that particular walrus in 2017, I believe it was also in August or September, that walrus was 5 times over the seafood safety limit for saxitoxin, which is the neurotoxin produced by the harmful species of algae called Alexandrium. So that was the beginning. At that time, we also had a mortality event, or a number of dead walrus. I think it was 29 or 39, several dozen walrus, again, from Wales to Espenberg. And those are very rare flights for us, especially being north of Nome like that. They take a lot of fuel. And so it was just sort of like, wow, okay. Now the carcasses in both cases were in a similar state of deterioration or decomposition, but we don't know when the death occurred, but all the animals were sort of in the same stage of decomposition. It wasn't like one had died here and one was fresh. They were all kind of the same level of rottenness. So
I guess what I'm getting at is I believe that these events have happened for a long time. The intensity of the events may be increasing, as far as the amount of saxitoxin being put into the water. I think the satellite evaluation of the water column is very important, but I also think the ice edge blooms are very important, and the actual phytoplankton blooms in the region are important to the benthos, and those animals may end up dying of starvation as opposed to toxins.
Correct.
I'm just pointing that out. I think it's great work.
Thank you, Dean.
Yeah.
Thank you, Dean. And I think that is really good to bring up in that there's a lot of focus right now and attention on the harmful algae, but we need to know all the algae. And I think with this satellite, that's what they're also trying to do. I mean, your focus is not just on harmful algae. You
That's
need to
right.
know when the satellite takes a picture. It'd be so wonderful, in my mind, listening to this, if we could get a satellite image, boom, hey, there's a problem over here, offshore. Ah, how nice. But they have that satellite, but they haven't got the how to read it yet. What does it mean in the water when they see their different images or the different 300 cuts of the purple, blue, and yellow? Beyond my
Yeah,
comprehension there. But
exactly. So actually the drone flights and also Mike's buoys, those are some tools that we want to focus on the spring bloom, which is usually not harmful.
Yeah.
But they can differ a lot in terms of diatoms versus the open ocean bloom.
Yeah,
versus
what's
the
out there?
ice
What's out there?
edge bloom. So
Yeah.
there are a lot of differences over there. So
we
hopefully
need algae.
yeah, hopefully the hyperspectral data can help with that.
Help understand our algae system, but also in the event that we did have something that was a species that is not good for
Yeah.
humans. So it's a bigger question of all the algae. And you
Exactly.
have this new tool up above us. So
Yep.
so I think it's awesome. This is a thing. What I noticed with harmful algae issues, or any algae issues at this point, is how much it brings everybody together. So your last slide where you had all the different, you need the community level, the research vessels, your aircraft, the satellite. Who did I leave out?
Surface buoys.
Surface buoys.
autonomous
Everybody comes together,
vehicles.
and it's very rare for me to see that level of teamwork for research in the ocean. It's really like the one ring to bind us all. Hate to say it, but that is really bringing people together in this region. So anyway, thank you for presenting that. Any
Thank you.
other? Oh, we've got some new messages.
I'd like
Better
to,
hush up.
can I say something in response to Dean?
Yeah,
Hi.
go ahead, Mike.
Yeah. Hey, Dean, I mean, I think you make good points, the satellite can't see below the surface, satellite can't see 10 years ago, but, or these new satellites can't, but, so what we got to do, we have to develop, if we can, proxies, right? We have to say, from what we can measure now at the surface, we can try to correlate behavior at the surface with subsurface behavior. We can try to correlate our new wonderful observations with old, less wonderful observations and try to get a time series, That's the best we can do at this point.
Is it possible to put one of these spectral systems that the satellite uses on one of your buoys?
Just
Just a question.
yeah, not at the moment. I mean, they're big and expensive and power hungry, So
Yeah, but there is a possibility that we can put this camera on the surface, the autonomous surface vehicles, for example.
yeah, a vehicle or a little drone, aircraft drone thing.
Yeah, definitely we need some power supply and some, bigger platform to carry that camera. So
Yeah.
Hello, Gay.
Yes, go ahead. Hello. Oh, and I don't know if we have a caller too. Callers should get priority on Street Science. I didn't even see that.
Oh, do you hear me now?
Go ahead. Yeah, go ahead, Edwin.
Yeah, if there's you talked about dead animals being washed up, and then you also if there's any cause on if they died of starvation or if they fed off of algae bloom that have full of toxins or whatever that had killed them. And you mentioned on some of the animals that were tested that there were high in saxitoxins, right? Or over
Correct.
the
The
limit.
one, correct. In 2017, there was a kind of an event that happened at Diomede that was late summer, early fall. I want to say August. This time, late August. And they were noticing several floaters go by, so dead walrus that were bloated. These were intact walruses, and because the waves were so large, people could not launch to go get those and you know, make use of the ivory. So that was kind of a note that interesting. 3 bulls went floating by. Then they had a walrus show up right out front, and people started calling them over because they wanted to harvest one. So they called them over, and the waves were getting down. Weather was getting better. This is secondhand, so pardon me on this, but this is how it was relayed to me. The walrus, there was a bull walrus, old bull walrus, that came out of the water, while they were calling, and it went underneath the corporation store, which is right over the beach. It's on pilings. And it went right up to it and put its head down on the beach in front of everybody. And for the next 45 minutes, it wouldn't raise its head. So Diomede was wondering, okay. So it looked like it had a behavioral problem. People wanted a walrus to harvest, so forth, so on. Anyway, the animal was dispatched, and then there was much more discussion about, well, wait a minute, are you sure you know? So I got called for, what samples do you need? We asked for a suite of samples, and actually got some intestine, the feces or unuk, tested in that walrus, and it was 5 times over the seafood safety limit. Is that what it takes to make a walrus act like that? So tired. Now, in human beings, this paralytic shellfish poisoning, we know what killed the walrus. A bullet killed the walrus, but the saxitoxin was 5 times over the seafood safety limit, meaning it had eaten something or a lot of something that had Alexandrium algae in it, ostensibly clams or whatever.
Can you detect that stuff in the fat or in the meat or just in the walls of the process?
So good question. So on this particular walrus, the feces lit up. Now, what kind of data does anyone have in the Western science world on the level of saxitoxin needed to make a walrus behavior go bad? Zero data, right? No one had ever seen this sort of thing. I mean, there have been some levels of saxitoxin detected in walruses, but no one has any idea on what it would take to actually affect the walrus's behavior. 5 times? That's a load, but we don't know.
Actually, we have Bill, who is
I understand.
expert.
I understand. We will get to, yes, I understand. We will get that. And Bill, you can correct me at any time. So as far as there's the EcoHab project, which is working with harvested animals off of St. Lawrence Island, of which I'm a part of that program, if that helps. And so is probably Bill. It looks like preliminarily, when we're taking a suite of tissues, when we're asking for a suite of tissues from the harvest, fat, muscle, intestine, unuk or feces, preliminary looks like muscle and fat or the blubber it's too slow to turn over that poison. So I don't want to say, I can't really say, you're going to be okay. But from the preliminary analysis being done by NOAA's Northwest Fisheries Science Center, Kathy LaFay, who's in charge of the EcoHab project, and if you want to, she's done several Strait Sciences, you can go back and see some of that. Preliminarily, it looks like it's very quick to get into the feces, but it is not quick to get into the muscle and the blubber. If that helps, Edwin, it would be a slower way for it to incorporate. What is it doing in the liver and the kidney? Whoa, we don't have those tissues. That's why the Fish and Wildlife is asking for suites of tissues from harvested animals, because they want to test all of it: liver, kidney, fat, muscle, and the unuk. Because if the unuk lights up that it has saxitoxin in it, you'll be able to test all those other tissues and say, well, we knew it had this much in the unuk. How much of that is in the intestine of the same animal? How much is in the liver, kidney, and so forth? Does that help?
Thank you very much, Gabe, for the explanation. Yeah.
You bet. Bill, do you have any anything
Bill,
to add on that?
Anything to add on that?
I not really. I think you summed it up really well, Gay, with our ability to detect these things is actually pretty low. So when we talk about like ability to detect impacts on walruses, for example, just because you don't see it doesn't mean it's not there. You know, that's kind of the point. We have to be a little kind of humble about our ability to detect these things, both in terms of the dose response, like how do HABs affect an individual walrus, and our ability to just opportunistically sample, right? If we sample 10 animals, right? Who's to say, you know, if we actually see 10 animals, who's to say how they died? You know, how do we get samples? So I think saying that, you know, that we would expect to see impacts right away is maybe selling our our ability to detect these things in wild populations a little too high. That'd be my take on it as a wildlife biologist.
So just to respond to that, if you don't mind, I'm jumping. Can I?
Yeah. Go
Okay.
ahead.
So for the USGS, which Bill Beatty works for the United States Geological Survey, which does the research on walruses and seabirds, the good work that's being done by USGS on captive seabirds, that's giving us more information on how saxitoxin works its way through the body on a bird. How you're right, we can't really take what happens to a murre and say that's how it's going to be for a walrus, but it does give clues. The other thing I would say is, even though you might say detecting impacts of HABs is, I would not say essentially non-existent. I will not be humble about the ability and knowledge base of our regional walrus harvesters and community members who live and work with walrus. The fact that people on Diomede immediately saw something that did not make sense. There's other biologists I've talked to that have seen walruses that are having trouble breathing as well. Those kinds of things, combined with the harvest, combined with the research, combined with the knowledge of the people of this region who live as you know, live with walrus and are the experts really on walrus comprehensively. I think that is going to be the key, right? The science, the knowledge, and catching those when the harvester may see something not right. I don't know. But I think we'll be able, as we get going more and more with sampling, I really do think that we will get better information on impacts. So don't give up. I'm ever hopeful. Thank you. Sorry, Jiaxu. We got off on walrus. So.
Yep.
To get a question?
Yes,
Go
to
ahead.
get us back.
Yep.
Yeah.
On like the second or third slide early, there was a map attributed to E.V. Vachon, and it said
Hello, Gabe?
2024. Wanted to just check, is that, so if we kind of go back, here's the start.
We're getting a question from one of the audience. I don't know if you guys can hear it yet. So when he's finished, we'll get to you, Ed, when I hear you.
Is that this?
Yeah, that one with, oh no, slide before that one. The chart in the bottom right, is that actually from 2024?
No, this is the publication that I got this image out of.
Got it. Okay.
So
cool.
the ship went there in actually the August. So I think there is it's August 17 to 31.
But of 2022.
But
okay.
2022.
Okay.
But the paper is out just a
Right.
few weeks ago.
Okay. Yeah, I was like, wait, is that this year? That's crazy.
Yeah.
So then the other thing I wanted to know about
Good eye.
is you mentioned the hyperspectral camera really just captures information at the surface of the water. But can you explain to me how the cube works? There's like a cubic, I think slide 4 there.
Yes.
Can the audience hear Ben's question?
Oh,
Yeah. So what I'm showing on the data cube is different from the actual depths that you're looking at. So physically, you're looking at the very surface or near surface layers, but you're looking at not only at the visual, like usually we see a RGB image, right? But now we're looking at all the different color information at this 2D location. So when I see this cube, actually, it's not on a depth dimension, it's on a spectral dimension that we're talking about.
Gotcha.
So
okay.
Yeah, this is a little bit confusing, but this actually, it's the spectral.
Gotcha.
Yep.
Cool. Okay, thank you. And if I could just add for people that can't hear this, that Pika camera is
You might want to
actually,
come on up here because I think we're
Well,
working off her.
it's just really small. I don't think people appreciate that camera is actually like pretty tiny when you see it in person.
Yeah, I probably can hold that thing, something like this big. I
Yeah.
would see. Yeah.
That's cool.
Yeah.
But
Okay,
so
thank
you fly high, so it actually covers, right? Quite a
Yeah.
bit.
If I fly, usually we do like 9,000 feet, and then the coverage, I mean, the swaths on the surface is about 4,000 feet. Like that's the width of it. But the lower you go, the narrower the swath is, but the higher pixel, I mean, the resolution is higher. So it's always a trade-off between resolution and the swaths.
You know,
So
I just
yeah.
remember when I saw it in the bay, you were like, and that's the hyperspectral camera.
Were you
I expected it to be like a much bigger thing, and then you pointed it out, and it was just like tiny, like the lens is like that big around.
yeah
And I thought it'd be like a giant,
yeah,
giant
there
camera.
are definitely some other like more professional. This is like a compact version, very small. There are other bigger ones with maybe bigger like better accuracy or, for example, better resolution. So they're all different kinds. There are definitely bigger ones out there. So
Cool.
Here's my question is, given the satellite, given the good Arctic air NOAA flying, given the ship based and everything's going, everyone's working together, at what point do you think we could just go, that satellite's going to go hook and we'll know, oh, this is that kind of algae, this is that kind of algae, no, this is good for us, we'll know the birds are going to get lots of food, whoop, that's the wrong kind of algae, we better be put out warnings. How long do you think that will be, or is it just that's what it's going to take forever, that kind of teamwork?
so good question. The picture that I paint for us, it will take at least 3 to 5 years
Okay.
to put all the component in. But I think that's also some question that Dean raised before, that does the concentration of the harmful algae mean toxin level? Maybe not. Maybe you have a good amount of, the harmful algae, the cell abundance is high, but maybe the toxicity level is not as high in the water. And also it depends on who is eating them and what's the timing of the eating and what's the path of this toxicity down the food chain, right, to the higher levels, the high trophics, the fish, the walruses, the seal. So it's all big question that our own program cannot answer. It's really the entire research community and also the local community will have to work together to figure it out. So yeah.
All right. Thank you very much for that. That's teamwork. That's Bering Strait. Any other questions, comments? Edwin, did you have another comment? And we have a caller, so if the caller just interrupt at any time. I've been bad to the caller.
It was, you answered, I think everything is pretty much answered. It was pretty good. So thank you guys. I'm going to leave. Yeah.
Okay. Next week for Strait Science, we will be having Strait Science on the Thursday, the 26th. And the speaker will be associated with the NGEE project. And that's a large scale project with Oak Ridge National Lab. So they've been working in the region for about 10 years or more, and they're going to give us the results. And their project expands from Nome, I believe, all the way to Utqiagvik. They have different stations going along, and they're looking at the environmental changes, mostly on land. So that will be next Thursday, and we'll get the invites out to all of you soon. Thank you so much. And if Mike, if you could stay on, and we'll have Jiaxu here for just a little bit after everybody leaves, that would be awesome. And shout out, Bill. Thanks for being there to help with the walrus. That's good. And we'll see you next time. Have a good night, everyone.